Class 7th, Subject General Science, Chapter 11, Cell Structure and Microorganism. Exercise. Question 1. Answer the following questions. A. What is the cell? Answer: the smallest structural and functional unit of an organism, which is typically microscopic and consists of cytoplasm and a nucleus enclosed in a membrane. Cells are the basic building block of all living things. B. Name the different organisms in a cell. Answer: these mainly include the nucleus, endoplasmic reticulum, Golgi bodies, lysosomes, mitochondria, vacuole, plastids, etc. Plant cells contain chloroplast. C. What are microorganisms? Answer: living organisms which cannot be seen with naked eye but visible under a microscope are called microorganisms. They are typically consist of only a single cell. Microorganisms include bacteria, protozoa, and certain algae and fungi. D. Which are the different types of microorganisms? Answer: microorganisms are divided into 7 types: bacteria, archaea, protozoa, algae, fungi, viruses, and multicellular animals, parasites. Question 2. Fill in the blanks with the proper words. A. The organelle, called the plastids, is present in plant cell only. B. Garbage is converted into fertilizer by microorganisms. C. In the cell, photosynthesis is carried out with the help of chloroplast. D. An electron microscope is necessary for the study of extremely minute organism. Question 3. What is difference between us? A. Plant cell and animal cell. Plant cell, animal cell. 1. Plant cell have cell wall. 1. Animal cell do not have cell wall. 2. Plant cell contain a single large vacuole. 2. Animal cells have small vacuoles. 3. Plant cells contain chloroplast. 3. Animal cells do not have chloroplast. 4. Plant cell have definite shape. 4. Animal cells do not have definite shape. B. Prokaryotic cell and eukaryotic cell. Prokaryotic cell, eukaryotic cell. 1. Prokaryotic cell are most primitive cells. 1. These are believed to have been evolved from prokaryotes. 2. They do not contain a definite nucleus. 2. They contain a definite nucleus. 3. Mitochondria are absent. 3. Mitochondria are present. 4. The chromatin bodies remain scattered inside the cytoplasm. 4. The chromatin bodies are enclosed by a nuclear membrane. 5. Prokaryotic cells are smaller than eukaryotic cells. 5. Eukaryotic cells are larger than the prokaryotes. Question 4. Sketch and describe in your own words the plant cell and animal cell? Answer: cell wall, chloroplast, cytoplasm, mitochondrion, vacuoles, endoplasmic reticulum, nucleus, Golgi body, cell membrane, lysosome, free rhizosomes, animal cell, plant cell. Animal cell. All animals are multicellular. All animal cells are eukaryotic. They are surrounded by cell membrane and do not have a cell wall. The components of animal cell are centrioles, cilia, flagelles, endoplasmic reticulum, Golgi apparatus, lysosomes, microfilaments, microtubules, mitochondria, nucleus, peroxisome, plasma membrane, and ribosomes. Plant cell. Plant cell are eukaryotic cell. They are membrane-bound organelles. They are surrounded by a rigid cell wall. Plant cells are similar to animal cell in being eukaryotic, and they have similar cell organelles. Question 5. Explain the uses and the harmful effects of microorganisms. Answer: uses of microorganisms. 1. They are used in the preparation of curd, bread, and cake. 2. They have been used for the production of alcohol since ages. 3. Commercially, microorganisms are used for the large-scale production of alcohol, wine, and acetic acid. 4. As a medicinal use, antibiotics are given which are prepared from microorganisms. Vaccines are another medicinal use of microorganisms. Harmful effects of microorganisms. Microorganisms are harmful in many ways. Some of the microorganisms causes the diseases in human, plants, and animals. These disease-causing microorganisms are called pathogens. Some microorganisms spoil food, clothing, and even leather. Question 6. Give reasons. A. Diseases spread on large scale during periods of heavy rainfall and floods. Answer: during the period of heavy rainfall and flood, the water and food get contaminated, due to which there is increased risk of water-borne disease. Diseases contract through direct contact with polluted water, such as wound infections, throat infections, etc. B. There is a possibility of food poisoning if we eat stall food. Answer: if we eat stall food, there may be a possibility that microorganisms produce toxic substance in food. So, when we eat such type of food, there is possibility of food poisoning. C. Soil is turned over during tilling. Answer: for proper mixing of useful bacteria in soil. Soil is turned over during tilling. D. Fungus grows quickly in moist or humid conditions. Answer: favorable conditions for fungal growth is 1. Moisture, 2. Humid atmosphere, 3. Slightly acidic pH. In moist and humid conditions, all such parameters are present, so fungus grows in such environment. E. A refrigerator is used in almost every home. Answer: if raw vegetables, fruits, milk, and many other things that are used in our day to day life. Kept outside, then it may develop a bad smell. Bacteria will spoil milk. All these are signs of spoilage. Hence, in order to preserve various things, a refrigerator is used in almost every home. F. Bread rises during baking. Answer. Breads are made of flour. Yeasts are added in the dough because they reproduce rapidly and produce CO2 during respiration, and so when it is added in the dough, CO2 is fills the dough and increase its volume. Hence, bread rises during baking. G. Fodder is soaked in water before offering to cattle. Answer. Fermentation takes place when we soaked fodder in water. Fermented fodder is easy to digest also. Fermentation increase in vitamin in the fodder, thus fodder is soaked in water before offering to cattle. Question 7. When will you use a simple microscope and when a compound microscope? Explain in detail how you will use them. Answer 1. Simple microscope is used at a basic level where there is no rigorous requirement of research. There is single lens in simple microscope. 2. Compound microscope has natural light source and mirror is concave reflecting type, whereas due to an added lens to a compound one, professionals use this for research purpose. 3. There is 3 to 5 objective lens in compound microscope which help in magnifying algae, fungi and bacterium. 4. In a simple microscope, the usage of hooks and knobs is not that much, where as in compound microscope the use of knobs in much, which help in focusing and as a result a clear and concise image in screen. 5. Compound microscope has a coarse adjustment screw for rapid focusing an object and fine adjustment screw for fine and sharp focusing. So it has wide range of use such as in studying the structure of different object. Example, detail of cell in living organisms.